Have you ever wondered how your body holds on to energy, not just for your next meal, but for that sudden sprint, or even just to keep your brain firing when you haven't eaten in hours?
Yeah, it's fascinating. It's not just about what you ate an hour ago.
Exactly. It's a remarkably sophisticated internal energy vault, constantly topping up and releasing reserves.
It truly is. Think of it as your body's primary quick access savings account for glucose, ready for immediate withdrawal.
And today we're taking a deep dive into that very vault, focusing on glycogen. This is the body's principal storage form of glucose.
That's the one.
And we'll unpack its intricate blueprint, understand its vital, well, distinct roles in different tissues, and unravel the complex dance of how it's built up and broken down.
And the regulation too, right?
Or
Because
yeah,
that's
yeah.
key.
Yeah,
Plus some clinical examples where things can go wrong.
absolutely. Our mission is basically to distill the essential insights from Chapter 26 of Marks Basic Medical Biochemistry.
Right, giving you a clear, accessible understanding of this fundamental biochemical pathway.
Think of it as your shortcut to appreciating why that stored sugar is so incredibly important for, well, everything from powering your muscles to sustaining your brain.
Couldn't have put it better. Shall we start with the structure?
Let's do it. So the blueprint. Imagine a highly branched, complex sugar molecule that's essentially glycogen, right? A branched polysaccharide made purely of glucose.
That's right. The individual glucose units are mainly linked by what we call 1-phenyl-4 glycosidic bonds, forming these long chains.
Okay, the main chain.
But the genius, really, of glycogen structure lies in its branching. Roughly every 8 to 10 glucosyl units, you'll find an unphenyl 1,6 branch point.
Ah, like a tree with many, many branches.
Exactly like a tree. And here's the key insight. This branching isn't just structural. It's a brilliant design feature.
How so?
It provides multiple active tips or non-reducing ends where enzymes can work simultaneously.
Ah, so it's like having many exits from a storage facility. Makes sense.
Precisely. It makes both filling and emptying the vault incredibly rapid, much faster than a single long chain would be.
Okay, and where do we find most of this stuff?
Well, many cells store a little glycogen, but the largest, most critical stores are definitely in your liver and skeletal muscle.
Liver and muscle, got it.
And these exist as massive, highly structured glycogen particles. There's even a little protein called glycogenin that acts as the initiator for each new chain, like a seed.
Interesting. So this amazing storage molecule, you said it has two very distinct jobs depending on location. Liver versus muscle. Let's tackle muscle first.
Okay, muscle. Muscle glycogen's purpose is pretty singular. Supply glucose 6-phosphate directly for ATP synthesis within that muscle cell.
So purely for the muscle's own use.
Right. Primarily through glycolysis. It's
Yeah.
a purely internal fuel, absolutely critical during intense exercise or when oxygen might be scarce, like in anaerobic conditions.
So if you're lifting weights or sprinting, your muscles are dipping directly into their own glycogen reserves.
Exactly. But here's a crucial detail that really defines its, let's call it selfish nature. Muscle tissue lacks a key enzyme, glucose 6-phosphatase.
Okay, and why is that enzyme important?
Well, without glucose 6-phosphatase, muscle cannot convert glucose 6-phosphate back into free glucose.
Ah, meaning it can't release glucose into the bloodstream for other tissues.
Exactly. It's entirely dedicated to the muscle's own immediate energy needs. It stays put.
Which brings us to liver glycogen. Sounds like it plays a different role entirely. More generous.
You could say that. Indeed. Liver glycogen is your first and most immediate source of blood glucose. Its job is maintaining stable levels throughout the body.
And that's critical because some tissues, like the brain,
Like the brain especially, yeah. They rely almost exclusively on glucose for ATP, and they need a continuous steady supply. The liver ensures that.
Okay, so how does the liver manage this trick? How does it release glucose into the blood when muscle can't?
It all comes down to that enzyme again. The glucose 1-phosphate from liver glycogen breakdown gets converted to glucose 6-phosphate, just like in muscle initially.
Right.
But then, thanks to glucose 6-phosphatase, which, remember, is present only in the liver and kidneys, that glucose 6-phosphate is converted to free glucose.
And that free glucose can leave the liver cell and enter the bloodstream.
Precisely, to go wherever it's needed, especially the brain, during fasting.
That's a fundamental difference then. And you mentioned fasting. This liver glycogen breakdown isn't working in isolation, is it?
Not at all. Especially during fasting or between meals, liver glycogenolysis is activated alongside gluconeogenesis.
Gluconeogenesis.
Yes,
That's making new glucose from scratch, basically, from things like amino acids.
Exactly, from non-carbohydrate sources. Hormones like glucagon coordinate these two pathways, glycogen breakdown and new glucose synthesis, to work together, ensuring your blood glucose stays stable even when you haven't eaten for a while.
Wow, okay. A very sophisticated system. So we have the structure, we have the distinct roles in liver and muscle. Let's dig into how it's actually made and taken apart. Glycogenesis and glycogenolysis.
Right, and what's really fascinating here, a core principle in metabolism, is that synthesis and degradation are rarely just simple reversals of each other.
Why is that?
By using different enzymes, the body gains independent on-off switches for each process. It's clever. It prevents this wasteful back and forth, this futile cycling, and allows for really precise control.
Okay, that makes sense. Let's start with building it up. Glycogenesis, how does that begin?
It kicks off with glucose getting activated. First, glucose enters the cell and gets phosphorylated to glucose 6-phosphate. This uses enzymes like hexokinase, or in the liver, glucokinase.
Okay, step one, phosphorylation.
Then, this glucose 6-phosphate is quickly rearranged into glucose 1-phosphate by an enzyme called phosphoglucosidase. Just shuffles the phosphate group.
And then it gets tagged somehow
Yeah.
for storage.
If that's a good way to put it. Glucose 1-phosphate then reacts with uridine triphosphate, UTP.
Yeah.
This forms uridine diphosphate glucose, or UDP-glucose.
UDP-glucose, okay.
This step requires energy from the UTP, and it creates this activated glucose unit. It's now primed and ready to be added to the glycogen molecule.
Got it. So once it's activated as UDP-glucose, what happens next?
The key enzyme glycogen synthase steps in. It takes that glucosyl unit from UDP-glucose and adds it to the non-reducing end of an existing glycogen chain, or a primer.
Via those ambulophore glycosidic bonds we talked about earlier.
Exactly, extending the chain. And remember glycogen, that
Yeah.
protein primer. It's essential here too, especially for starting totally new chains through a process called autoglycosylation, basically adding glucose to itself before glycogen synthase can really take over.
Right, so glycogen synthase makes the chains longer.
Yeah.
But what about the branches?
Good point. Once a chain gets long enough, maybe around 11 glucosyl units, a different enzyme called the branching enzyme, its technical name is amylose 576 transferase, performs its magic.
What does it do?
It cleaves off a chunk of the chain, maybe 6 to 8 residues long, and reattaches that piece to the side of the chain using an al-1,6 bond.
And that's how we get those crucial branches.
That's exactly how. Increases solubility and, importantly, creates more non-reducing ends for enzymes like glycogen synthase and the breakdown enzymes to work on simultaneously. More ends means faster action.
Brilliant. Okay, so that's building it. Now, the flip side, breaking it down, glycogenolysis. Where does that start?
Breakdown begins with the main enzyme glycogen phosphorylase.
Okay.
It works at those same active non-reducing ends, but instead of adding glucose, it breaks the I-1,4 bonds. And it does this using inorganic phosphate, a process called phosphorylasis.
Not hydrolysis, phosphorylasis, so it adds a phosphate group.
Correct. It directly releases glucose 1-phosphate, so most of the glucose comes off already phosphorylated.
Does it just chew all the way down the chain?
Not quite. This enzyme actually stops about 4 glucose residues away from an I-1,6 branch point. It gets kind of blocked, sterically hindered.
Ah, so it leaves these little stubs near the branches.
Exactly. And that's where the debranching enzyme comes into play. This is a really cool enzyme because it actually has 2 distinct activities rolled into one protein.
2 activities, what are they?
Well, first, it acts as a transferase. It basically shifts a little block of 3 glucosyl residues from that stub over to the end of a longer nearby chain, reattaching them with an I-1,4 bond.
Okay, so it tidies up the branch, mostly. What about the single glucose unit left at the branch point?
That's its second activity. It acts as an A1,1-furazix-glucosidase. It uses water hydrolysis this time to clip off that single remaining A1-filial-6-linked glucose residue.
And that comes off as free glucose.
Yes, as free glucose. So for every branch point broken down, the overall yield is mostly glucose 1-phosphate plus 1 molecule of free glucose, roughly a 7.1 or 9.1 ratio depending on the branching density.
Interesting. So you get both forms.
You do. And just briefly, there's also a minor pathway involving lysosomes. A lysosomal glucosidase can also break down glycogen, and this becomes really important when we talk about certain diseases.
Okay, good flag. Because these pathways aren't just, lines in a textbook. When they go wrong, the impact is real and can be profound. Let's shine a spotlight on some of those clinical cases, starting with Gretchen C, the newborn with critical hypoglycemia.
Right, Gretchen's case. It's a stark illustration of just how critical liver glycogen is, especially for newborns. She was delivered emergently because her mother had severe appetite loss late in pregnancy.
So the mother wasn't getting enough nutrients.
Exactly. And at birth, Gretchen was tiny, limp, cyanotic, bluish skin, and her blood glucose was dangerously low, only 14 milli-GDL.
No.
Normal for a newborn should be over 40. She quickly became unresponsive.
That sounds terrifying. What's the direct link to glycogen here?
Well, during the last 10 weeks or so of gestation, a healthy fetus actively stockpiles glycogen in its liver, using glucose from the mother. Gretchen's mother's malnutrition meant Gretchen simply didn't build up adequate stores.
So she was born with an empty fuel tank, essentially.
Pretty much. After birth, the cord is cut, the maternal glucose supply stops. Normally, the baby's own hormones, glucagon, epinephrine, kick in to mobilize that stored liver glycogen to keep blood sugar up.
But Gretchen couldn't do that effectively.
Right, her reserves were too low. This led to profound hypoglycemia, starving her brain and causing those life-threatening symptoms. Timely feed glucose saved her, but it really highlights how vital those initial glycogen stores are.
A really powerful example. Okay, let's shift gears to case number 2, Jim B., the young bodybuilder.
Jim. A very different scenario, but also highlighting glycogen and glucose control. He was a 19-year-old bodybuilder, rushed to the ER. He was in a coma, having grand mal seizures.
Also low blood sugar.
Extremely low.
Yeah.
His serum glucose was 18 milli-GDL. He later admitted he'd been using anabolic steroids, but also injecting large amounts of insulin before his intense workouts.
Injecting insulin, why would a bodybuilder do that? I thought that was for diabetes.
Well, insulin does promote glucose uptake into muscle and can increase muscle glycogen synthesis, which some bodybuilders misuse thinking it boosts muscle growth or performance. But it's incredibly dangerous.
Clearly. So what happened?
He created a perfect storm for severe hypoglycemia. The massive insulin overdose would have forcefully driven glucose from his blood into his muscle cells. Crucially, insulin also strongly inhibits glycogen breakdown in both the liver and muscle.
So it locks glucose into storage and prevents release.
Yes, and it stops the liver from making new glucose via gluconeogenesis too. So his liver couldn't release any glucose into the blood. Then he started exercising intensely.
Burning through whatever glucose was left in his blood.
Exactly. His muscles were using up the remaining blood glucose rapidly, and the liver couldn't compensate because the insulin was blocking release. His brain, starved of its essential fuel, shut down, leading to the seizures and coma. Aggressive VA glucose was needed immediately.
Wow. A stark warning about misusing hormones. These individual cases, Gretchen and Jim, they sort of lead us into a broader category, glycogen storage diseases, or GSDs.
Right. These are a group of inherited genetic disorders, each caused by a defect in one of the enzymes involved in glycogen synthesis or breakdown. They really highlight specific points where the system can fail.
Can you give us a couple of examples?
Sure. Let's take Pompe disease, which is type 2 GSD. This is caused by a defect in that lysosomal α-glucosidase we mentioned briefly earlier.
The minor pathway enzyme.
Yeah. Without it, glycogen builds up inside lysosomes, particularly in liver and muscle cells, eventually disrupting their function. It used to be fatal, especially the infantile form, but thankfully, enzyme replacement therapy is now available and can be life-saving.
Okay, what about another one? Maybe affecting muscle more directly.
Good example is McArdle disease, type V GSD. This is a deficiency in the muscle form of glycogen phosphorylase.
The main breakdown enzyme in muscle.
Correct. So the problem isn't storing glycogen. Muscle can store it just fine. The issue is mobilizing it during exercise. Muscle glycogen cannot be broken down efficiently for energy.
So patients would get tired very easily during exercise. Cramps.
Exactly. Severe fatigue, exercise intolerance, and painful muscle cramps are classic symptoms. It primarily affects skeletal muscle, so while debilitating, it's not typically life-threatening like some other GSDs.
And one more, maybe affecting the liver. You mentioned von Gierke disease earlier.
Right, von Gierke disease or type I GSD. This one is particularly severe. It's caused by a deficiency in that key liver enzyme, glucose-6-phosphatase.
The one that lets the liver release free glucose.
Precisely. Without it, the liver can make glucose-6-phosphate from glycogen, but it can't convert it to free glucose to release into the bloodstream. So glucose-6-phosphate accumulates inside the liver cells.
And what does that accumulation do?
Paradoxically, high levels of glucose-6-phosphate actually push pathways towards more glycogen synthesis, even when the person has low blood sugar, hypoglycemia. So you get this massive buildup of glycogen in the liver, leading to a hugely enlarged liver, or hepatomegaly, and very severe fasting hypoglycemia because the liver just can't do its job of maintaining blood glucose.
That sounds incredibly complex to manage.
It is. Requires careful dietary management, often continuous glucose supply.
Okay, so given all these potential problems and the different needs of liver and muscle, it just underscores how critical the regulation of glycogen metabolism must be. How does the body keep it all straight?
Absolutely. The regulation is elegant and precise. The overarching principle, as we touched on, is that liver and muscle glycogen pathways are controlled to meet the specific needs of each tissue, and critically, to prevent that wasteful, futile cycling breaking down and building up at the same time.
Makes sense. Let's start with the liver again. Hormones seem key here, right? Insulin, glucagon.
Very much so. Think about after a meal. Blood glucose goes up, insulin levels rise, and glucagon levels fall. Insulin is the dominant signal here.
And what does insulin do in the liver?
Insulin essentially flips the switch towards storage. It triggers a cascade that activates a key enzyme called protein phosphatase 1, PP1. This phosphatase removes phosphate groups from two crucial enzymes.
Okay.
It dephosphorylates and therefore activates glycogen synthase, the building enzyme. And it dephosphorylates and inactivates glycogen phosphorylase, the breakdown enzyme.
So synthesis on, breakdown off, store the glucose.
Exactly. Net effect.
Yeah.
Glycogen synthesis is stimulated, degradation is inhibited, perfect for storing that post-meal glucose surge.
Okay, so what happens, say, hours later when you're fasting and blood glucose starts to drop?
Now the hormonal picture reverses. Low glucose means low insulin and high glucagon. Glucagon becomes the dominant signal for the liver.
And glucagon does the opposite of insulin.
Pretty much.
Yeah.
Glucagon triggers a different signaling cascade, this one involving cyclic AMP, CMP, and protein kinase A. pKa adds phosphate groups. It phosphorylates things.
Okay, so phosphorylation turns things on or off here.
Right. pKa phosphorylates and inactivates glycogen synthase, building off. And it phosphorylates and activates another kinase called phosphorylase kinase, which in turn phosphorylates and activates glycogen phosphorylase breakdown on.
So synthesis off, breakdown on. Release the glucose.
Precisely. Glycogen degradation is stimulated, synthesis is inhibited. The liver starts releasing glucose into the blood to maintain levels.
And what about epinephrine, the fight-or-flight hormone? Does that affect the liver too?
Yes. Epinephrine F also strongly stimulates liver glycogenolysis, getting glucose out quickly for an energy surge. It actually works through two different receptor pathways in the liver.
Two ways.
Yeah. Through beta receptors, it activates the same CAMP-PKA pathway as glucagon. But through alpha receptors, it triggers a different pathway involving increases in intracellular calcium, Ca2+. And calcium also helps activate phosphorylase kinase.
So both pathways push towards breaking down glycogen.
Exactly. They work synergistically with glucagon, if it's present,
Yeah.
to maximize glucose release during stress or exercise.
Wow. And there's even a direct effect of glucose itself on the liver.
There is, yes. It's quite neat. When blood glucose levels rise rapidly after a meal, glucose itself can directly bind to the active form of glycogen phosphorylase, phosphorylase A in the liver.
And what does that binding do?
It makes the phosphorylase a better substrate for that protein phosphatase 1, PP1 we mentioned earlier. So it promotes its dephosphorylation and inactivation.
Ah, so it's a very rapid shutdown signal for breakdown, even before insulin fully kicks in.
Exactly. It provides a very quick initial break on glucose release as soon as glucose starts arriving from the gut.
Very elegant. Okay, let's switch focus to skeletal muscle glycogen regulation. You said it's different because its needs are different.
Exactly. Muscle glycogen is all about providing ATP for the muscle itself, so its regulation reflects that local need.
What are the key differences from the liver then?
Several big ones. First, glucagon has no effect on muscle glycogen. Muscle cells don't really have glucagon receptors.
Okay, so fasting signals from glucagon don't tell muscle to break down its glycogen.
Correct. Instead, muscle responds strongly to signals of its own energy status. A key signal is AMP adenosine monophosphate. When ATP is used up quickly during intense activity, AMP levels rise.
Indicating low energy charge in the cell.
Right. And AMP acts as an allosteric activator of muscle glycogen phosphorylase. It binds directly to the enzyme and switches it on, stimulating glycogen breakdown right when the muscle needs more fuel.
So it's a direct feedback loop within the muscle cell.
A very important one. Another key signal is calcium, C2+. Remember how calcium triggers muscle contraction?
Yeah, released from the sarcoplasmic reticulum.
Well, that same rise in calcium during contraction also directly activates phosphorylase kinase in muscle.
The enzyme that activates glycogen phosphorylase.
Correct. So the very signal that initiates contraction also simultaneously boosts the fuel supply pathway by stimulating glycogen breakdown. It synchronizes fuel delivery with demand.
That makes perfect sense. Any other muscle-specific points?
Yes, unlike the liver, glucose itself is not really a physiological inhibitor of muscle glycogen phosphorylase. Muscle breakdown is driven more by AMP and calcium. Also, glycogen itself acts as a stronger feedback inhibitor of muscle glycogen synthase.
Meaning when muscle glycogen stores get really full, it slows down further synthesis.
Exactly. Helps prevent the muscle from overstuffing itself with glycogen.
But epinephrine still affects muscle, right? Like in fight or flight.
Oh, yes. Epinephrine is a major activator of muscle glycogenolysis. Just like in the liver, via that beta receptor, CMP, PKA phosphorylation cascade, it provides that rapid burst of glucose 6-phosphate needed for glycolysis during sudden exertion or stress.
Okay. This regulatory network is incredibly intricate. Before we wrap up, you mentioned an enzyme, glycogen synthase kinase 3, or GSK3, that has broader implications.
Yes, GSK3. It's a fascinating story. It was initially discovered, as the name suggests, for its role in inhibiting glycogen synthase by phosphorylating it at multiple sites.
So it puts the brakes on glycogen storage.
Initially, that's what we thought its main job was. But it turns out GSK3 is a real cellular multitasker. It phosphorylates something like over 60 different proteins, maybe more,
Wow. Doing what?
influencing a huge range of cellular processes: cell structure, motility, growth, survival. It's often involved in pathways where it acts on substrates that have already been primed by phosphorylation from another kinase. So it's often a downstream regulator, integrating signals.
Okay, so it's way more than just a glycogen enzyme. Does it still connect back to insulin and glycogen?
It absolutely does.
Yeah.
One of the key actions of insulin signaling through a pathway involving ACT, or protein kinase B, is to inactivate GSK3.
So insulin shuts down the enzyme that inhibits glycogen synthesis.
Exactly. By inactivating GSK3, insulin relieves the inhibition on glycogen synthase, allowing it to become active when dephosphorylated by PP1 and promote energy storage as glycogen.
And this has clinical relevance beyond glycogen.
Definitely. Because GSK3 is involved in so many pathways, its dysregulation is being implicated in major diseases. For example, in type 2 diabetes, a loss of insulin's ability to properly inhibit GSK3 might contribute to insulin resistance.
Interesting.
And perhaps even more surprisingly, GSK3 activity has been linked to the pathology of Alzheimer's disease, potentially relating to the phosphorylation of tau protein. This makes GSK3 a really hot target for drug development, although its widespread roles make it tricky to target specifically without causing side effects.
Really illustrates how interconnected everything is at the biochemical level. Well, what a journey that was. We've navigated the intricate branched structure of glycogen,
Explored its very distinct and vital roles in the liver versus the muscle.
delved into the detailed steps of building it up glycogenesis and breaking it down glycogenolysis.
And witnessed the really elegant orchestration of its regulation by hormones like insulin, glucagon, epinephrine, and even internal signals like AMP and calcium.
And understanding this delicate balance, it's clearly not just academic, is it? It's absolutely vital for appreciating those real-world conditions.
Exactly, like the critical hypoglycemia in newborns like Gretchen, or the, frankly, dangerous misuse of insulin by someone like Jim.
And it's fundamental for diagnosing and managing metabolic diseases, the glycogen storage disorders, type 2 diabetes.
Absolutely, they all hinge on understanding these pathways.
And as we consider the sheer complexity here, and especially the far-reaching influence of enzymes like GSK3, it really makes you wonder, doesn't it?
How many other pathways are hiding similar complexity?
Yeah. How many other seemingly specific biochemical pathways hold such crucial keys to understanding broader physiological processes, maybe even future therapeutic targets? What other hidden connections might we uncover next in our body's intricate designs? It's quite mind-boggling.
It really is a constant source of discovery. Thank you for joining us on this deep dive into the world of glycogen.